our next session of In the Pipeline. So now we're talking about fibrosis and inflammation. So Eric, can you give us a little explanation about what's ahead in this panel?
Absolutely. So as Ryan mentioned, we're going to be talking about some strategies that are combating fibrosis and inflammation. We've had a number of these discussions now as we lead into these In the Pipeline sessions about all of the issues that can arise when we are lacking that dystrophin protein, which is so foundational. When we lack that protein, those muscle cells that are damaged, they have impacts in their ability to repair properly. Over time, that damage can lead to replacement of the muscle tissue with fat and scar tissue, and that process is known as fibrosis. And so what we are looking to do with some of these therapies are trying to slow that progression of fibrosis and slow that disease progression as well. So by addressing a number of different factors, whether that's inflammation or muscle regeneration or directly targeting different fibrotic factors, we can look to decrease fibrosis, we can look to improve muscle function, and really try to provide some benefit. So we are going to be joined today by Dr. Bassem Elmankabadi from FibroGen, Dr. Paolo Bettica from Italfarmaco, and Dr. Linda Marban from Capricor Therapeutics. So as a reminder, as we go through these sessions, if you have questions, please submit them in the comment box below, and we will try to get to as many of those in the Q&A as we can. And so for this session, we are going to start off with FibroGen.
Hi, Eric. Thank you for the nice introduction. And I'm glad to be presenting today on behalf of FibroGen to PPMD. And I want to thank the committee and PPMD and Pat Forlong for inviting us to participate in that session. With that said, so our program in Duchenne muscular dystrophy, we are starting phase 3 trials called Lelantus study, and we are led by Dr. Khosrowshahi. He is our senior VP in clinical development and drug safety, and myself and Ken Lepson, our executive director in drug discovery. We had a presentation, I believe, in the website posted there. So next slide, Eric, please. All right, so first introduction. Who are we? We are, and then I'm going to go discuss the phase 3 Lelantus trial, design, endpoint, and inclusion-exclusion criteria in non-ambulatory patient. And then I will going to give you a heads up about our upcoming also ambulatory study in study 094 Lelantus 2. And then I'm going to conclude my presentation, and then I'm going to open the floor for question and answers. Next slides, please. All right, so FibroGen is a mid-sized biotech company. We are located headquartered in San Francisco, California, and we are around $4 billion company. And FibroGen has 2 platforms, anemia roxadustat, which is under review for FDA for approval, and anti-fibrosis pamrevlumab, which is a monoclonal antibody. Pamrevlumab is being studied as we speak in Duchenne muscular dystrophy, idiopathic pulmonary fibrosis, locally advanced pancreatic CA, and COVID-19. Next slides, please. Right now, I'm going to discuss the phase 3, the upcoming phase 3, which is currently started phase 3 studies, and it's called Study 93, Lelantus, and I'm going to review the design and the study period in brief. It's a global randomized double-blinded trial of pamrevlumab or placebo in combination with systemic steroids in subjects with non-ambulatory Duchenne muscular dystrophy, age 12 and older. It's 90 patients, male, of course. Subjects will be randomized at 1-to-1 ratio to arm A, pamrevlumab plus systemic steroid, arm B, placebo plus systemic steroid. The screening period will be for 4 weeks, up to 4 weeks. Treatment period for 52 weeks, last dose at week 52, and then safety follow-up period will, and the end of the study is 1 month extra, which is a 20-day plus minus 3 days. So this diagram here, or the table here, has arm A and arm B. Arm A is pamrevlumab 35 milligram per kg, intravenous infusion bi-weekly, plus systemic steroid or deflazacort or inflaza, any equivalence potency for corticosteroids. And arm B, matching placebo, intravenous biweekly plus systemic steroid, deflazacort or inflaza, administered, of course, orally. Subject will be the subject who complete the 52 weeks of the study in either arm may be eligible to roll over to an open label study extension of pamrevlumab plus systemic steroid, in addition to any approved therapy of Duchenne muscular dystrophy, to be on board with pamrevlumab in the OLE section. Next slide, please. So that's a summary of our primary endpoint and the secondary endpoints, and those are based on the new guidelines by FDA that has been published in 2018. The primary endpoint will be the function assessment, will be the change in the total score of the performance of the upper limb using version 2.0 from baseline to week 52. The secondary endpoints will be pulmonary, three buckets or three categories: pulmonary assessment, performance assessment, and cardiac assessment. The pulmonary assessment divided into two parts: the change in percent predicted forced vital capacity from baseline to week 52 assessed by spirometry, change in percent predicted peak expiratory flow rate from baseline to week 52 assessed also by spirometry. The performance assessment will be the change in the grip strength of the hand from baseline to week 52 assessed by handheld myometry. And then the last one, which will be the cardiac assessment, will be the change in the ejection fraction from baseline to week 52 assessed by MRI. Next slides, please. And then this is the second phase 3 trial. The first one that I just discussed is the phase in non-ambulatory trial, study 093. And this is the second trial, phase 3, that we're going to start by Q4 of this year. Will be an ambulatory trial, Duchenne muscular dystrophy, of course, and this is ambulatory patients 6 to 12, randomized multi-center double-blinded trial. The randomization will be 1-to-1. We are planning to enroll 70 patients. Arm A will be the pamrevlumab 35 milligram per kg intravenous biweekly plus systemic steroid or deflazacort administered orally. In the arm B will be the matching placebo plus steroid. Randomization will be stratified by the gene 44 exon skipping. And we are aware that the gene 44 skipping has the late onset of ambulation. That's why we are planning to certify that. Subject who completed the 52 weeks of the trial in both arms will be eligible also to roll over into the open label extension, where pamrevlumab plus systemic steroid will be given in addition to any approved therapy of Duchenne. Next slides, please. And this is a heads up of the primary and secondary endpoints. And again, based on the FDA guidelines for industry and published in 2018, primary endpoint would be the North Star, change in North Star from baseline to week 52. Secondary endpoint will be four-stair climb velocity assessment from baseline change from baseline to week 52, and the change in 10-meter walk-run test from baseline to week 52, and the changes in the time to stand from baseline to week 52. And I believe that's about it. And I'm going to open for any questions. Thank you, Eric.
Thank you, Dr. Elmankabadi. I have the first question before we turn it to the next speaker. With your ambulatory study that you'll be rolling out, given sort of the progress that we've seen with imaging DMD, I'm wondering if you all are considering doing MRI, skeletal muscle, in your trial to track that over time for the ambulatory population.
Absolutely. That's an excellent question. And we do have an expiratory endpoint. I don't want to put it in the slides here because of the timing of the presentation was 6 minutes only. However, yes, the answer is yes. We have MRI for vastus lateralis muscles, which will be analyzed and assessed by a blinded charter for imaging charter based on the guidelines by FDA. And it will be an expiratory endpoints in that trial to measure the fibrosis.
Wonderful. Okay, thank you so much, and we'll see you in the Q&A panel. Our next speaker from Italfarmaco is Paolo. Hi, Paolo.
Good to see you.
Good to see you as well. Please take it away. It's your turn for your presentation.
Okay, thanks a lot. Well, thank you, Ryan. Thank you, Eric. And obviously, thanks the whole PPMD organization for allowing us to actually tell you where we are in the development of givinostat. So if I can have my first slide first, please. Okay, excellent. As usual, my disclaimer. So I'm a full employee of Italfarmaco, which is manufacturing givinostat. Givinostat is currently in development, so it's not available commercially in any country, including the USA. And obviously, this presentation is intended for dissemination only and discussion of scientific information. So next slide. As Eric actually summarized very, very well, we know that there are some key important events that occur once dystrophin is not present, as is the case in Duchenne. One is that the muscle is unfortunately going into more frequent damage, but then also the other issue is that the repairing system actually does not work properly. And as a result of that, instead of replacing muscle with new muscle fibers, the muscle is actually being replaced with fibrosis, with scar, or with fat. So the way we believe that givinostat works is actually on the repairing system. So it works by reducing inflammation, it works by reducing fat and fibrosis replacement, and it works by increasing regeneration. So if you go to the next slide. So if you take all together what I just said and try to understand which are the potential benefits of givinostat, well, what we believe and what we have shown in a couple of publications is that givinostat is actually counteracting the events which are triggered by the lack of dystrophin that eventually lead to muscle replacement and to all the functional loss that is you are very well aware of. And as I said, we have shown this in preclinical studies, and we have shown this also in a phase 2 study in DMD boys, where after 1 year of treatment of givinostat on top of corticosteroid, we have seen an increase in muscle fiber diameter and in regenerative fibers, and a decrease in tissue damage, inflammation, and fat replacement. Actually, those kids that started taking givinostat a long time ago, more than 6 years ago, are still on treatment. So we can now look at what givinostat does on top of corticosteroids, for example, on the morphology of the muscle, so on fat infiltration, looking at that via MRI, and we see that there is less fat infiltration compared to an external control. We see a delay in disease progression, for example, age at loss of ambulation, and we also see a delay in respiratory function decline. So if you go to the next slide. So this is a summary of our phase 3 study, which is called the EPD Study. The objective here is to demonstrate that givinostat preserves muscle mass and delays disease progression. And to do so, we are actually measuring a number, we are performing a number of tests in the boys who are in the study. The primary endpoint is actually the 4-stair climb, but then we are measuring other functional tests, the North Star, the time to rise, the 6-minute walk test that you are very well aware of. We are measuring strength. As Ryan was mentioning before, we believe that MRI is actually very important to look at the evolution of the muscle measured directly by MRI, and this is actually a significant component of our study. And also we are measuring pulmonary function and heart function. So in the bottom part of the slide, you see the scheme of the study. The kids are firstly screened, and also an MRI is being performed during the screening. Then, if they are eligible for the study, they are being randomized 2-to-1 to givinostat and placebo, so they have more chance to actually getting givinostat. And then they are being treated for 18 months total. And during these 18 months, they actually attend the clinic for a number of visits. These are usually more frequent at the beginning, in the first 3 months, to ensure that we define the best dose for the kid. And then they are followed up every 3 months. They are also going for an MRI scan at 12 months and 18 months. And then once the study is completed, they can enter the open label extension study, which I'm going to describe in a minute. As I said, there are a number of visits, especially at the beginning. We are trying to help the families and the kids by actually having nurses going to their home when only a blood sampling is necessary. We are actually helping the families for traveling in terms of both arrangement and of reimbursement. And as I said, at the end of the study, they can move into the open label extension. If you go to the next one, this is a brief description of the open label extension study. So the kids are actually taking givinostat. Every one of them is taking givinostat. The objective here is to look at the safety and tolerability, but we are also looking at the function effect in the long term. And here the visits are usually every 4 months. And on the right-hand side, you see the different tests that are being performed, which are very similar to the one that I just described before for the EPD study. So if you go to the next one, here is where we are. Part of the EPD study was an interim analysis done by an independent data monitoring committee. The objective there was to decide whether we had sufficient result to continue enrollment, and the answer was actually yes. So the study is continuing. And also we actually looked at the variability of the test of the forced air climb to see whether we could potentially readjust the sample size to ensure that we had the proper power for the study. And the good news is actually the analysis showed that we can reduce the sample size to actually 169 subjects. So where are we? Actually, today we are at 166. So we are actually very close to the end of recruitment, and hopefully that could actually be met by August and not even September. And obviously the final data will be available in the second quarter of 2022 after 18 months of the last patient. And as a result, then the estimated submission in the US and in Europe is actually for 2023. So if you go to the next one, just to tell you that Italfarmaco is actually working also in Becker. We are actually conducting a phase 2 study in Italy and the Netherlands. The recruitment is complete and we'll have results next year. And also we have other molecules actually in the preclinical phase that hopefully will also help in Duchenne. And if you go to the next, just really to thank obviously all the families and boys that are in our studies and all the associations that have been helping us. And at the bottom, you see actually a link where you can actually find more information regarding the studies. Thank you.
Great, Paolo, thank you so much. Just one follow-up question. We saw in your slides the mention of the Becker studies that you have done, and those have been in Europe. Are you planning a phase 3 there? And importantly, this is certainly a high unmet need for clinical trials within the U.S. Are you looking to make it global, and will the U.S. be included in such a study?
So at the moment, the plan is actually to wait for the results of the phase 2. And obviously, based on the results of phase 2, then if they are positive, certainly there is an interest in actually expanding the development.
Wonderful. Okay, great. Well, we will be waiting for news, and we appreciate you. You're always communicating with us and with the community, and we'll invite you to a webinar at any point to have a further discussion. Okay, thank you. So our next speaker is Dr. Linda Marban. Hi, Linda. She's going to give us an update from Capricor. Linda?
Hi, thank you so much. Thank you to Parent Project, and thank you for putting together this great meeting during these unprecedented times. During these unprecedented times, I will give you fair warning that I am home and it is a Saturday, and so my grandchildren are here. So if you hear noises in the background, they're not pre-recorded. So I'd like to talk to you today about CAP-1002 and our program in muscular dystrophy, of which I am extremely proud to present. Next slide, please. We're a publicly traded NASDAQ-listed company under the symbol CAPR, and all of our filings are up to date with the SEC for your review. Next slide, please. So let me tell you a little bit about CAP-1002. CAP-1002 is a cell therapy. It's an allogeneic off-the-shelf product. We make our cells from donor hearts, so they're a very unique cell type, but they are not a stem cell. They don't work like stem cells do. They don't go into tissues and take hold and all end up multiplying and making new ones of themselves. What they do is they release these very tiny particles called exosomes, and exosomes contain all kinds of proteins and nucleic acids in order to send messages between cells. And so what they primarily do is they have an immunomodulatory capability and then also a pro-regenerative capability. The immunomodulatory capability is based on the fact that not only is inflammation slowed, but it also allows the cytokines that are good for making muscles repair themselves to be activated. This then drives muscle cells back into the cell cycle, which is how new ones are made, to make new healthy muscle cells. We've been studying these cells for about 15 years. We started off in treating heart disease primarily and moved into muscle, primarily in Duchenne muscular dystrophy, because we realized that the cells could work not only in dealing with the cardiac implications of Duchenne, but also the skeletal muscle implications of Duchenne. The cells have been in over 150 people, and we have a very strong safety record. Next slide, please. So let me tell you a little bit about our clinical work in Duchenne muscular dystrophy. And many of you have heard me give webinars before, and I talk a lot about the preclinical data that led to these clinical work and clinical studies. But today I'm going to focus mostly on the clinic. So we focus on the later stage non-ambulatory DMD patients. 80% of our patients in both of our clinical trials have been non-ambulatory. And so we focus on upper limb function and then, of course, the cardiac function and respiratory function. The first trial that we did, because we were still trying to target only the heart disease, it was we infused 75 million of the cells into the coronary arteries of patients. And these were 25 patients, 13 got cells, and 12 were standard of care. And what we found were several very important things. One, we found improvement in measures of cardiac function, and we also found improvements in measures of skeletal muscle function as measured by the performance of the upper limb 1.2. All of this work was published in the Journal of Neurology in February of 2019, and it led to a transformation of our program, which has led to us to where we are today. One was we realized we needed to dose with more cells. We got an impact, but needed a greater impact. Two, we realized from preclinical studies done during the clinical trial that we can move from infusing cells directly into the heart, requiring a visit to the cath lab, to infusing cells directly by intravenous infusion. So a simple IV delivery takes about half an hour, and patients sort of can be outpatient. They're monitored to make sure that they're safe, and then they're allowed to go about their days. And this is a quarterly infusion. Next slide, please. So as I mentioned, Capricor is primarily focusing on the later stage patients, those with upper limb function morbidity already, that is impairment in their ability to use their shoulders, their arms, and their hands. Shown here is a schematic of the performance of the upper limb, ranging from a score of 0 in a task, which means you have no ability to perform that task, to 6, which means you completely can perform that task and have absolutely no inabilities at all. We focus primarily on pull entry scores of between 2 and 5. That means you still have room to improve, so we can see if CAP-1002 is working, but you also are not so healthy in that measure that we can't measure improvement. We hope to at some point move to the ambulatory population, but our focus has been on the huge unmet need for those that are non-ambulatory. What we've learned primarily from working with Parent Project, which are all of you, is that the upper limb function is really the gateway to maintaining human privacy and independence. And so we want to see if we can have a therapeutic that takes us in that general direction. Next slide, please. So now let's talk about HOPE 2.0. That was the clinical trial that we've just completed, and we are in discussions with FDA at this point. We're looking to see if we can qualify for some version of accelerated approval because the data that we found were extraordinary. Shown on the right are graphs of individual patients that were treated. It was a small trial, 20 patients, but the results show the profound implications for patients. This is the performance of the upper limb 2.0 full, so shoulder, arm, and hand. And what we see is that there's a 2.3 point difference between those that were treated with CAP-1002 over the course of 1 year, 4 infusions, compared to placebo patients. And the placebo patients really got better or did not get better. They sort of followed the trajectory that's been published many times. We usually use the work of Marika Payne as our guideline if you want to review the literature. So natural history was the same as our patients, and a 2.3 point preservation of function. So this gave us really a journey that we've completed now with Eric Henriksen at UC Davis, where we've looked at the clinical meaningfulness of these scores. And to put it into context, the data shows using Synergy, a data set, and then a lot of natural history data, that even a 1 point change would have a clinically meaningful difference. Next slide, please. And we saw incredible improvements in cardiac function. I say incredible improvements because I'm a cardiac physiologist by background. And so when we see changes in volumes and ejection fraction over the course of a year, we realize that we have something very unique and very special here. So remember, this was a doubling of the dose from HOPE 1, where we saw a little bit of change in cardiac function. Now, after a year, in a blinded core lab, the images were read by Dr. Conhor, who I think most of you are aware of at Ohio State. And he had no idea patient identity, and we saw improvements in ejection fraction, how the heart meets the needs of the body, and also changes in the shape of the heart, helping it go more back to a normal shape and dimension so it can meet the needs of the body. The heart is like a physical organ, not just part of your body, but physics control it. So pressures and volumes regulate how it functions. So the more normal the shape, the better you are at meeting the needs of your toes and your brain. Next slide, please. So where are we going with this? Well, we're very excited with this data. There's a lot more of it, and I showed it on your webinar, but I can also make it available if you're interested. We're working with FDA now. We've put in a proposal for accelerated approval. We'll see how that goes. It's a small sample size, but we've met every other one of their requirements, and we're also trying to encourage lobbyists and legislators to talk to FDA about if you have a safe therapeutic during the time of COVID, it would be great to make it available. Open label extension starts very soon. Imminently, the patients will be treated. So if you were in HOPE 2 and you want cells, they are now available to you, whether you were a placebo or a treated patient, for 4 doses. The cells were mostly safe. In general, we had 1 or 2 reactions where patients needed to be brought back from an allergic response, not brought back like they died or something like that, but they had an allergic response, and we had to attenuate that. We now pre-treat with a very successful pre-treatment regimen. It's oral. It's very easy for the patients to take. We watch them for an hour or 2 afterwards, make sure they're okay, and everybody is up and on their way. The one patient that had the event of an anaphylactoid type response is just fine now. Okay, so I think that's all I've got. Next slide, I think, is there a conclusion slide? Do I have one more? I don't remember. Nope. Okay, so this is my last slide. So please feel free to ask me any questions or let me know how we can help you get better.
Great. Thank you so much, Linda. Really appreciate that. One quick question before we open it up to the panel Q&A. If you do receive approval, do you anticipate any issues with manufacturing?
Right. So we're in a manufacturing partnership with Lonza. So we're in late stages of that. And we anticipate that we will not have any of those issues by the time we get to commercial stage. So the goal will be to have doses ready from our Lonza manufacturing either by the end of the OLE or by the time we prepare the BLA. So all things working as they are now, we don't anticipate any issues.
Excellent. Thank you so much. All right, Eric?
Yeah, so
Q&A.
We are going to open it up for questions. And we had a great one come in already. And this is going to go around to the whole panel from a parent. And it's discussing that, the fact that these drugs tackling things like fibrosis, scar tissue, fat deposition, they're going to be helpful not just as monotherapies, but taken with potentially some of those exon skipping gene therapies. So their question was, first of all, why aren't we seeing combination trials for these type of drugs? And then just as a follow-up, do you have plans to evaluate your investigational products along with any exon skipping or gene therapy products? Paolo, I'm going to actually start with you.
All right. Well, thanks for the question, which is a recurrent theme because it's very logical and very sensible. So why are we testing our drug as monotherapy? In reality, because this is the first step that is being required by the regulatory authorities. So the regulatory authorities are requiring as a first step to clearly show what is a benefit and what is a risk-benefit ratio of your single drug. So that is a necessary step. Having said that, I think what is being asked is absolutely logical. We are all, I think, fully convinced that combination treatment will be very helpful in this disease. And clearly, once we have shown that there is an effect of our drug, that will be certainly a next step to be considered very thoroughly.
Great. No, thank you for that explanation. I think that really does help families kind of understand what that process looks like. Bassem, are you potentially going to explore the use of your product with exon skipping or gene therapy?
Yeah, I concur actually with Paolo, and I believe that's because of the agency's request. It's a monotherapy is mandatory, at least for phase 3, to be for consideration for BLA or NDA. And yes, the answer is yes. We are in the OLE extension. We will going to give pamrevlumab at least for FibroGen. We're going to give the FibroGen drug plus Exonadis or Vyondys or any approved therapy with pamrevlumab in the same time. So the answer is yes. We are trying to study in the open label extension the combination therapy between pamrevlumab as an antifibrotic in addition to any of the approved therapies.
That's great to hear. Thank you. And Linda, same question.
Yeah, so we already allow patients to be receiving exon DYS51 or any of the approved exon skipping drugs. And gene therapy, absolutely. We can't do it for the same reasons that everybody else said experimentally. And mostly we're also treating the non-ambulant kids. They don't qualify typically for the gene therapy, so it hasn't been a competitive issue. We have done preclinical work with the gene therapies that are available, some of the microdystrophin therapies, and have been able to show that the cells work just fine and the gene therapies work just fine independent of each other. So we're poised to do that when the time is right.
Okay, great. Thank you. This one for FibroGen. Do you anticipate any delays to that Q4 start for your ambulatory trial this year with the impact of COVID?
Yes, the answer is I don't want to say anticipate a delay. We are working on it, and the protocol has been submitted to the health authorities already, and the sites are in the process of to be selected. And we had a lot of progress in the non-ambulatory trial. However, for ambulatory trial, we just started like a month ago, and I hope that we're going to make the timeline by end of this year. So I'm optimistic, and I think we will going to make it.
Great. Thank you.
Can I ask a question?
Yeah, go ahead.
Yeah, so first of all, I want to recognize for all of you, all looking at trials and have been doing trials in populations of unmet needs, non-ambulatory and Becker. We appreciate that. The community appreciates that. So I just wanted to throw that out there really quick. For Linda, I wanted to ask about, with seeking accelerated approval, you're seeking this on non-ambulatory, I'm assuming, of course, because you're going for non-ambulatory, but what about ambulatory populations with your drug? Are you hoping for a broad label at the FDA, or what's your thinking around this for ambulatory?
Well, it's a sticky question anyway. We're pushing and we'll continue to fight for the accelerated because we believe that patients deserve the opportunity for this. We've seen really relevant improvement in functions and clinically meaningful ones, and we'd much rather see boys and young men in local infusion centers than on airplanes 4 times a year to go to another clinical trial site. But the FDA has not given us any reason to believe that they're going to approve that, so I don't know. And we'll sort of go, my general concept is we'll go with the narrower label, should we be lucky enough to convince them to let us go ahead for our non-ambulance, and then expand into the ambulant population over time. But I don't know the answer to that.
So thank you. I appreciate the answer. Thank you.
Yeah. We had another parent asked, Paolo, do you expect Givinostat to have the chance at reducing the amount of steroid that someone is able to take if they're taking both Givinostat and a steroid?
So this is actually a good question, and it's certainly something that we want to look at in the future. There are obviously a number of commonalities actually between Givinostat and steroids. And so the possibility of actually reducing the dose of steroids needed is certainly something that we want to explore in the future.
Great, thank you. And as a follow-up as well, I had a question about whether or not we could expect to see any of the interim analysis data that you discussed earlier, if that would be shared.
So actually, the interim analysis at the moment is unknown to us also, because it's done by an independent data monitoring committee. Remember, the study is double-blind, so we cannot absolutely jeopardize the study by actually showing the results. Once, obviously, the study is complete, then all the results will be shared with no problem.
Okay, great, thank you. So we do have to take this time and wrap up, so thank you all for participating in this call today.